there are still people coming in, but I don't want to shortchange our speaker. So maybe I will wait a minute or so. I see a lot of people coming in. Come on up, there's seats up toward the front. So welcome everybody. This is, I believe, the last in our series this year, the Pioneers in Biomedical Research series, so thanks for coming out. We'll be gearing up again in the early fall with the beginning of our series, so watch the website for the list of speakers that we'll have in the coming year. I'm sure it'll be equally as exciting as the program we've had so far this year. So it's a great pleasure to introduce today's visitor to the Virginia Tech Trillium Research Institute, Dr. Michael Failings, who has a medical degree and a PhD. He currently serves as professor of neurosurgery and vice chair in the Department of Neurosurgery for Research. He also holds the Halbert Chair in Neural Repair and Regeneration, and serves as the co-director of the Spine program at the University of Toronto, where he also heads up the Spinal program as the Irvy's Senior Scientist at the Toronto Western Research Institute and the Toronto Western Hospital. Dr. Failings did his PhD at the University of Toronto, working then on the role of calcium channels in experimental models of spinal cord injury, an area of focus that he has followed in many directions over the years. He then went on and completed his medical degree also at the University of Toronto, and then while there at a series of the affiliated hospitals, did his neurosurgical residency training. And I'm not quite sure I understand how they do the residency programs there, but it looks like you did residencies in neuroradiology and neurosurgery and neurology, but I gather that's all part of the entire training. Yeah. And from there, of course, joined the faculty. He has a number of awards and recognition, and one that I find very interesting that I haven't talked to him about yet, I'll say it now, that creates an interesting little connection. He's a recipient of the H. Richard Wynn Research Prize of the Society of Neurological Surgeons. So I don't know if you know this, Dick Wynn trained at the University of Virginia in Charlottesville, and I was training in the lab next door to him, and we worked together for about 6 months, a long time ago. And I did a variety of projects with him and then followed his career out in Seattle, and it's great to see that you were honored by the Dick Wynn Prize. He's also received a variety of other prizes, the Henry Farfan Research Award of the North American Spine Society of the United States, the Medal for Spinal Cord Injury Research, and the American Academy of Cerebral Palsy, and the Developmental Medicine Research Award. He has been recognized as a fellow of a variety of societies, the Royal Society of Canada, the Canadian Academy of Health Sciences, and the American College of Surgeons. He serves in a whole bunch of capacities in terms of editorial boards and service that I won't go through. And his research has taken, I think, a fascinating, converging approach to a very big problem. He can certainly tell us about the impact and prevalence and incidence of spinal cord injury. I think everybody in this room is aware of that impact in terms of numbers, economic cost, and burden that it has on individuals and their families. It's, I think, one of the great challenges that we face of really trying to figure out how to repair the damaged central nervous system. It's a multifaceted problem, and Dr. Failings has come at this with basic science approaches, with translational approaches, with clinical approaches. It really is the whole package. He has done important work on cell-based repair systems for spinal cord injury, investigating the role of transplantation, for example, of neural stem cells. He has continued on with his interest in the role of calcium signaling and understanding the role of different types of receptors, such as metabotropic glutamate receptors and calcium signals in axonal dysfunction in the injured spinal cord, and even gone as far as looking at things like cytoskeletal degradation, cell death, and all the important biological processes, channelopathies, et cetera. But again, more importantly, he scales back up to the patient and the entire problem. Has been involved in a number of very important trials and studies bringing this information together, and really represents, I think, the best in many ways of the translational medicine approach to a big problem. So please join me in welcoming our guest, Dr. Michael Failings.
Oh, thank you. Wow. I don't know if I can live up to that introduction, but that's very kind, and it's an honor for me to be here. And it's my first time to Roanoke, and it's a very beautiful community. I've been learning about your wonderful research institute, so thank you again for the honor to be here. So I'm going to take you on a little bit of a journey, and from molecule to man. So that's a rather presumptuous title. But this is kind of what I've been trying to encompass. And truthfully, probably about 90% of what we know about research and the management of traumatic spinal cord injury has been learned in the last 2 decades. And what we're now witnessing is a remarkable era, and this is the era where we're starting to see the translation of discoveries relevant to repair and regeneration of the injured nervous system into one of the most devastating forms of central nervous system, namely traumatic spinal cord injury. So I have no commercial disclosures to declare, and this is some of the sources of grant funding. So this is where I live and work. So this is Toronto, and I work at the Toronto Western Hospital, which is one of the big teaching hospitals in the University of Toronto system. And usually you thank all the members of your lab and your collaborators at the end, but I like to do this at the beginning because really the work that I'm going to present is very collaborative in nature and have a wonderful team of collaborators and students. My mentor, Dr. Tatter, is on my right, and he's 80 years young, and he's really the reason that I went into spinal neurosurgery and did my PhD work. And the young woman who's sitting in front of us, that's Barbara Turnbull. She was injured at age 18 as a result of a gunshot wound at the C2 level, and she's sort of been a local heroine and local mascot. She actually went on to become a journalist and tragically died last year at age 50, which is about the average lifespan for somebody with a severe cervical spinal cord injury. And so while we can save people's lives, spinal cord injury is a life-threatening condition that dramatically shortens people's lives. So I'm just going to begin this with a clinical vignette. So I'm a practicing neurosurgeon. I take care of people, and I got a call a few summers ago from one of my friends who's a neuroradiologist at the University of Toronto. They were vacationing in Northern Ontario, beautiful lakes there, and unfortunately his friend dove into shallow water. He's 39 years of age, and this is what a cervical fracture dislocation looks like. So this is a CAT scan, and the 4th cervical vertebra is dislocated in front of the 5th cervical vertebra. This is a magnetic resonance scan that was done shortly thereafter, and shows that the spinal cord's been crushed. Spinal cord's rarely transected. It used to thought that was the actual mechanism. It's not. It's a physical contusion and a compression. There's black signal in the level of the spinal cord here. This represents hemorrhage. And then this white signal that's going up into the spinal cord, and below this, this is edema, and this is what's referred to as a secondary injury. And so this man has an injury at the C4-5 level, and as a result of this, has what's referred to as a C4 level ASIA A injury. And this means that he has no motor or sensory function below the level of his injury. He can barely shrug his shoulders. He has poor head control. He has absolutely no trunk control. He has no volitional control of any of the functions below the level of his shoulders. So this is an absolute nightmare for this patient, but this is also a life-threatening injury because the signal change is going up to the C2 level, and this is where you breathe, right? This is where the phrenic motor neurons are located. So there's nothing that I can do that's going to reverse this. We can do things, though, to save this man's life, to try to get small improvements in function, and that is important. But what else is out there, and what are some of the emerging approaches? So I'm going to talk to you about this, and I'm going to start with maybe telling you a little bit about the story of spinal cord injury and this concept of the secondary injury. And I'm going to talk to you a little bit about some of the clinical issues and some of the struggles I've had in terms of trying to develop evidence-based protocols. And it can be a challenge, even when you have evidence, to try to see the incorporation of this. And I'm going to talk about some of the areas of research that are going on, including areas that others are doing. Spend a little bit of time on regenerative neuroscience, and then come back to the clinic with a clinical trial that we're going to be starting pretty soon with the C3 row inhibitor, and this is a regenerative strategy. So here's another young person I looked after, not as severe. He's got a traumatic fracture dislocation at C6-7. This is a mid-sagittal section. This is a post-mortem. This is from a young person who succumbed from the injury, and it highlights that we can think about traumatic spinal cord injuries involving a two-step process. There's an initial physical trauma. So this is what's referred to as the primary injury, and it's pretty easy to see how this occurs. So there's tremendous physical violence that occurs. The spine is dislocated. There's hemorrhage in the disc. There's contusion, compression. But there's also a set of events that occurs which amplify the nature of this primary injury. And this has been referred to as a secondary injury. And you get a little bit of a hint of this, that there's some hemorrhage that's occurring in the spinal cord. And when I was doing my PhD work, there was huge pushback against this concept that there was a secondary injury. And there's what I might refer to as a sense of therapeutic nihilism. And what I would say to all of you is that when somebody tells you can't do it, you should just take that as an incentive to mean that what you're doing is really interesting and you should actually focus on that and to do that. So I was told that you're absolutely wrong, Failings, you're crazy, your methods are poor, and there's no such thing as a secondary injury. And in fact, it turns out that the work that we had done was validated. And so this was one of the pieces of evidence. This is from an animal model, and we've used what's called a clip compression model. And later on the talk, I'll show one slide where we've done this. And here we've injected the blood vessels. This is colloidal carbon microangiogram. And this is anterior spinal artery, and the vessels arborize like on the branches of a tree. And then there's hemorrhage that occurs, and this is due to a primary mechanical damage. There's physical disruption of these blood vessels. There's hemorrhage that occurs. But what's interesting is that when you look around here, this area looks relatively intact, and this is what's been referred to by Lindsay Simon as the ischemic penumbra. And this is the penumbra, it's in the shade. And so these nerve cells and nerve fibers in this area are alive, but they're ischemic. And if you image these, and here we've done confocal immunohistochemistry for a molecule called NF200, we see that initially these axons are intact, and then they undergo secondary axotomy. And this is due to the influx of molecules such as calcium and sodium, and there's a secondary degradation of the cytoskeleton due to activation of molecules such as calpain. And then this culminates in the neuropathology of a human spinal cord injury, and this is shows here in the chronic phase. So this hemorrhage gets resorbed, there's loss of tissue, and this results in a cavitation or myelomalacia. And this is the astroglial scar, and this is very important in limiting the extent of injury, but tragically it has the unintended effect of limiting the regenerative response due to upregulation of molecules in the extracellular matrix, including chondroitin sulfate proteoglycans, and then there's an anterograde and a retrograde degeneration of these axons. And so you didn't start off with a transection, but you end up with a virtual transection. And each one of these points represents a separate pathobiology and separate strategies. And so this is kind of a simplistic concept of some of the positive feedback loops that are involved in the secondary injury cascade. And we had proposed what was fairly radical concept as early as 1992 when we published our paper on the ischemic hypothesis of secondary injury, which is now essentially become fairly well accepted. And so we feel that ischemia is probably the key trigger, and then there are changes that occur at the macromolecular level and also at the cellular level. And initially, some of the critical events that occur are related to loss of homeostasis of the cell membrane with influx of sodium, and this triggers a secondary activation of calcium influx, and there's a whole host of ionic shifts that occur, release of neurotransmitters, and secondary apoptosis and inflammation. And so this sets really in motion these positive feedback loops. And there's a lot of investigation that's going on right now in terms of identifying other molecular targets. But this is, we know enough now about these secondary injury mechanisms that we can actually translate this into clinical therapeutics and potentially into sensible clinical trials. So this is a cartoon that one of my graduate students put together kind of summarizing some of the strategies. So here we have the lesion. We have the lesion epicenter. It's walled off by the glial scar. We have demyelination of some of these residual axons, and I'll show you how this occurs later. So we have this primary mechanical injury, and then we can think about methods to potentially protect some of these nerve cells or potentially even to replace or regenerate these nerve cells. So let's talk about surgery. So I'm a surgeon, and recall this patient, and I showed you images of how the spine is compressed. So does it actually make sense that surgery might have a role, and we might actually be able to influence the trajectory of outcome? And there was this huge sense of therapeutic nihilism. We talked a little bit about this last night at dinner. Why isn't everybody having surgery right away? Because it was a feeling that it doesn't really matter. And so I decided that, well, okay, if I was going to try to translate some of these concepts of secondary injury, maybe we should look at this. And for sake of time, I'm just going to cut to the chase. We did a multi-center international trial called the STASCUS trial, which is a surgical time and acute spinal cord injury study. And we found that, in fact, when we operated within the first 24 hours after injury, and in fact, sooner is better than later, that the odds of improving the outcome significantly was almost threefold higher. And then there's been subsequent meta-analyses of these, which I've also shown this, and then there's been subsequent prospective studies that have validated this. And this is another Canadian cohort study that showed improved outcomes. So where this is now going is in terms of the development of guidelines. And this is one group that I belong to. AO Spine has close to 20,000 members worldwide. And this is we're trying to adopt this at internationally level, that early surgical decompression should be offered as the preferred treatment option for patients with acute spinal cord injury. And when you're talking to clinicians, you're trying to influence outcome, words matter in the way you word this. So you see the way I've worded this is that should be, and it's a preferred treatment option. And this is to avoid, medical legal concerns and this sort of thing, because you do have to individualize patient treatments depending on the individual patient scenario. This is from the cover of an internal medicine journal read by primary care physicians and internists, and I felt that it was important to communicate some of the information, say, around time of spine and around some of this information, not to surgeons, but to people that are engaged in primary care or are dealing in the emergency rooms, because there has been this sense, well, it doesn't really matter when you refer a patient with a spinal cord injury, and yes, of course, it's a devastating injury and it's tragic, but it doesn't really matter when you do this because you can't really do anything. I felt that was really important to counter that and to also point out that small differences make a difference. So with that individual, I pointed out, right, he's got a devastating injury, and I'm not going to be able to return that person to what he had before. But if I could get a little bit of hand function back in that individual, right, that would have a huge impact in that individual. And so small differences matter. And so here I just outlined sort of, and this made the cover of the journal, and I just sort of outlined in a time course some of the early events that would occur around the concepts of early surgery and early intensive care that make a difference, and then was talking about some of the early phase trials, and I'll talk to you about some of the strategies we're talking around there. And remarkably, currently in the United States and Canada, and you're going to find this amazing, but spinal cord injury is not being triaged as an emergency. Okay, it's not. There's not the concept that this is a time-dependent manner, and this is why we've evolved this concept of time of spine. The other problem that I see creeping up all the time is what I call therapeutic nihilism. So I debated whether I would actually include the next few slides, because I think this is probably mainly a bit more of a basic science audience, but I think you need to understand some of the challenges around translation. And this also kind of deals with human nature, but also something that I refer to as therapeutic nihilism that you really have to watch out for. So there was a big randomized trial that came out. It was published in 1990 by Bracken et al., and then there were 6 other randomized trials showing that methylprednisolone, which is a very powerful corticosteroid and immunosuppressant, resulted in modest gains in improvement in spinal cord function. And then there was a little bit of a backlash against this, primarily for medical legal reasons, because not all physicians were using this. And so as a result of this, there was a lot of bad feelings. This was reviewed at a guidelines level in 2002, and despite the fact that there were these randomized trials, it was felt that, you know, this can't really be a standard of care because the effect sizes were small and was recommended as an option. And you know what? This was probably reasonable to recommend, so it allows physicians the option. But unfortunately, therapeutic nihilism crept in, and a decade later, in the absence of any further big trials, they actually said it's not recommended. And it was felt that, you know, the effect sizes are too small, and it's a hassle to give, and there's the cost, even though the drug doesn't cost very much, and the average spinal cord injury results in lifetime costs of between $2 and $20 million. So a cheap spinal cord injury is around a $2 million injury. So like in my country, this is a multiple billion-dollar expense for the healthcare system, and you can multiply that by the usual tenfold difference between Canada and the US. This is a big issue here. Okay, so what changed to warrant this recommendation? And so I felt that I kind of couldn't stand by and watch this. And so this is what's called the evidence tree. This is how we evaluate levels of evidence in medicine. And so you've got, like me, talking up their ideas, opinions. Okay, that's at the bottom here, right? And then you've got kind of case series, and you start coming up here, case series, but they're controlled, prospective cohort study, you start getting to the randomized controlled trials, and then you get to systematic reviews and meta-analyses. So these is where you don't just have one randomized trial, you have several randomized trials, and you synthesize the evidence. That's felt to be the highest level of evidence. And to have a recommendation, like a strong recommendation or a guideline, you need to have level 1 evidence. And this is really only based on systematic reviews or randomized trials. And that wasn't the case with methylprednisolone. So why did I feel that was important? I don't have any commercial interest in methylprednisolone. The concern that I had was that if the medical community wasn't going to accept the results of well-done randomized controlled trials that had a small effect size, because the effect sizes were small, we're dead in the water. We might as well stop trying to advance anything in the field of spinal cord injury. In fact, you could probably end translational neuroscience, because the therapeutic effects that we're going to witness are probably going to be small. It's unlikely that any of us are going to come up with the silver bullet that's just going to, be able to make all of the phenotypic presentations vanish. Small differences matter, right? And therapeutic nihilism is to be avoided. So I thought, okay. So what we ended up doing through this not-for-profit foundation is we decided that, okay, we're going to have an external group of methodologists review all of this and just, okay, let's just put an end to this. So they did this. This is what's called a forest plot. And so a forest plot is when you take the results of all sorts of different trials, and you use very sophisticated statistical algorithms to bring the data together, and you have the positive trials, and you have the negative trials, and so on. And this is kind of the bottom line. So here, this is where there's no effect. Negative means that it favors the control, and this favors methylprednisolone. And so what we found was that, sure enough, taking all of the data together, that there was a small effect that was favoring methylprednisolone. So this is about 4 to 5 motor points of recovery when we score these different motor points. So that means that someone might be able to recover, say, the use of their hand, or they might recover some grip strength. It means that on average, patients, aren't kind of, having a huge transformation, but there are small effects. And then we looked at safety issues, and we found that although this is a very powerful immunosuppressive drug, there was no negative effect on survival. In fact, it slightly favored the drug, probably because of some of the beneficial effects. And then we took some of the data from my clinical trial, the STASCUS trial, and then we tried to look at complications, and we actually found that steroids had reduced complications, probably because the patients were being benefited We then had the data vetted independently, and we found, sure enough, that there was reduced risk of complications. So that's the first step that you do. So you do what's called a meta-analysis. You do systematic reviews, and you bring the data together. So that's kind of that aspect of it. But the development of guidelines actually have to take that evidence, and you now have to filter this through expert opinion, and you have to take into factors. How important is the effect? How feasible will this be to administer? Is it affordable? Does it matter to patients? Is it important? What is the risk and the benefit? So if you have something that has some benefit, but it's so risky to take forward, you're not going to want to do that. So that was one of the arguments against corticosteroids. Oh, it's so risky and the benefits are less. But when we actually looked at it, we said, well, okay, this is what the data show, and this is what the effect sizes are. And so we put together a multidisciplinary group. This is one of the meetings that we had in Toronto. It's not pictured here, is we actually included people who were in wheelchairs. So we had a couple of people in wheelchairs. We had some people that were from the NGOs, including the Christopher Reeve Foundation. Because when you're starting to ask questions like how important are these effect sizes, it's one thing to ask a physician, but it's another thing to actually ask somebody who's in a wheelchair. And then you then have to kind of look at the balance of consequences, and you have the type of recommendation. And so, and then what this is actually a pretty rigorous process. I'm just kind of getting to the bottom line, but it was felt that the desirable consequences probably outweigh the undesirable consequences, and we suggest offering this option. And then there's a lot of discussion that was going around this, and we were looking at the heterogeneity of the injuries, and then we're trying to make some clinical recommendations in terms of where this might actually have an impact in people who have a cervical injury, and particularly if you combine this with early surgical decompression. And so this I think will alter care, but it's also important because there are trials that are going on now. You know, I agree that the effect sizes with corticosteroids are a bit disappointing. I would like to see larger effect sizes, but we're talking about a therapy that was developed in the 1950s and the 1960s, and we're debating a clinical trial that was published 26 years ago. That's really quite remarkable. So this is a short summary of some of the neuroprotective drugs that are currently in trials, and these two are currently active, and I'll talk to you about the Riluzole trial, which is a trial that I'm leading. Minocycline is a trial going on with an anti-inflammatory drug called minocycline. GCSF is a pro-inflammatory cytokine. This is in a small phase trials in Japan, and the group in Miami is proposing to look at systemic cooling. And then these are two early phase 2 trials that were actually aborted. These were industry trials. Remarkably, the two industry trials were stopped because I think industry was concerned around some of the issues I raised, that, you know, if we come up with a positive result, will the clinical community come back and say, oh, it's not very important? And so what's the point? These 4 trials are being moved forward by academia in conjunction with peer-reviewed funding and NGOs. So here's a strategy that we're moving forward with the sodium channel blocker, Riluzole. We're studying patients with cervical injuries, and here I'm modeling what's called a burst fracture. And then we have the primary injury, and then this is amplified by the secondary injury cascades. So if we now look at the nerve cells and nerve fibers, here I'm modeling the node of Ranvier. We have voltage-gated sodium channels, and there's a rapid influx of sodium through these channels. Then there's a reverse operation of the sodium-calcium exchanger. The cell tries to pump the sodium out, but calcium comes into the cell, and you have a pathologic release of glutamate, and there's a failure of sodium-dependent glutamate uptake mechanisms. So this triggers some of the classic events that occur in early phase secondary injury. And then what we're now studying in clinical trials is optimizing early medical and surgical care. So we're doing the decompression, we're stabilizing the spine. The concept is to optimize the milieu for recovery, and then we're giving Riluzole to try to improve the outcome. So to do this kind of work, you have to start with a strong basic science background, but then you need to filter this through an expert group to try to decide which drug are you going to move forward, and then one needs to have a consortia. So I'm fortunate to belong to a couple of consortia, one of which is the North American Clinical Trials Network, and this is funded through DoD and the Reeve Foundation in partnership with AO Spine. So this was the rationale for moving forward with this. So Riluzole is the only approved neuroprotective drug in the world. It's available all around the world, and it's used in a condition called ALS, or amyotrophic lateral sclerosis. And we don't know the cause of ALS. We don't know why the nerve cells degenerate, but we do know that it involves some of the events that I've described in spinal cord injuries: sodium and calcium influx, glutamatergic excitotoxicity, oxidative cell stress. So Riluzole is currently a standard of care to try to slow down the rate of nerve cell degeneration in ALS. And so we felt that we had proof of concept in men in a related condition, and we had safety. But we still felt that we needed to do an early phase, a trial, and that was published. And we found that the drug had a good safety profile, but we also found that there was a significant improvement in neurological function. This is the Asia Impairment Scale grade. This is a way that we grade changes in function. Asia A are patients who have no motor or sensory function below the level of injury. Asia B have some sensory preservation. Asia C have some motor recovery. And then these are the conversion rates going from one level to the next. And we found that Riluzole improved the conversion rates by about 25%, and this was significant. And then we also found that the trajectory for motor recovery, particularly in patients with higher injuries in the cervical spine, was improved. And so this is right now in a phase 3 multi-center randomized trial. The trial's been launched. We have 45 patients that are enrolled. This is by far the hardest trial I've ever run. It's extremely challenging. We're dealing with traumatic injuries. The patients are sick. We have a relatively narrow time window to give the drug, within 12 hours. But I'm quite hopeful. We'll see. Now, there's other things that are out there, and I wanted to give you a sense of a few of these things. These are two strategies that are not looking at regenerating or neuroprotection, but these are taking advantage of some of the underlying circuits in the spinal cord and the brain. So this is some interesting work from Harkema and Edgerton's group. And here what they did was they took electrical stimulators that are used clinically to treat neuropathic pain. It's called a dorsal column stimulator. And they applied them to the lumbar spinal cord of people who are paralyzed with injuries in the neck and upper thoracic spine. And the lumbar central pattern generator is like a computer program that has the circuits that can trigger movement. And if you give sensory stimulation, you can trigger movement. And so these are EMG records in an individual who is totally paralyzed, and they found that they could evoke movements in the legs. And this was published in Brain a couple of years ago. So they're now in early phase clinical trials with this. So it's really cool to think that maybe you could somehow, even if somebody's paralyzed, you might be able to evoke movement. And you may have seen this paper. This was an N of 1. This was a application of brain-computer interfaces. So how does an N of 1 get published in Nature? Well, because of the rigor of the way the work was done, and it was really, it's very disruptive in a positive sense. And Ali Rezai was actually trained in our unit in Toronto. He's now at Ohio State, and this is an individual who had a complete cervical spinal cord injury, not too dissimilar to the patient that I presented. They identified with fMRI the motor cortex. They applied, they recorded the signals. They amplified these through a computer interface. This is showing the forelimb, and they were doing the recording here, and they were able to get significant movements here with the person thinking about moving the limb and this being transferred to the forelimb. Like, really cool, really cool. Okay, so I'm going to come back here to some of the challenges of spinal cord injury. So the reality here is, even with early surgery, even with a neuroprotective drug, the person that I presented at the outset, they're going to be left with a very disabling situation. And there are going to be people in which it's really worth it to think about a regenerative therapeutics. So here I'm coming back to the human neuropathology, the challenge of spinal cord injury. So we have a loss of neural cells. There's clearly disruption of circuits, and there's a discontinuity of the axon tracks. And some of the axons that maybe are preserved, they're very demyelinated, so they don't transmit the signals properly. And you have a physical cavity, right, which is a barrier, and then there's the glial scar, and then you have the expression of inhibitory molecules. So what can you do with cells? And so these come in all sorts of different flavors, but the reason that people have been using these different types of cells is because it's been really hard to get neural precursor cells. And the approach that we took was we said, we're going to try to go with neural precursor cells anyway, and maybe we'll figure out a way to make these, because our feeling was that if regenerative technologies were going to have a role, we might want to go with the optimum cell type. So instead of going with the easy cell type and seeing whether it might have a role, we actually went with the most difficult cell type. And the cells can work in different ways. So you can think about potentially replacing the cells, but they might provide trophic support, release of growth factors, modification of the environment. They might actually immunomodulate the environment. And all the central nervous system has been thought to be relatively immune privileged. It's not as immune privileged as you might think it is. And so in fact, the inflammatory cascades are important. And then one might actually, these cells might actually promote an intrinsic scaffold. And so I sort of put this slide together, just to share with you kind of some of the thinking that I've been going over in the last 10 years in our own laboratory to try to figure out, okay, how are we actually going to do this? And this is really kind of inspired by some of the patients I'm treating. So I'm focusing on cervical spinal injury, thinking about a translational pathway. So the first thing was, can we even get these neural stem cells to work in the optimal models? And then can we figure out a way to derive these from induced pluripotent stem cells? And we have a non-viral way to make these iPS cells. Can we come up with a translationally relevant cervical spinal cord injury? It didn't exist, mainly because there were high mortality rates in the cervical spinal cord injury models. And then can we figure out a way to get human iPS cells to work here, and can we overcome the xenogenetic barriers in terms of studying human cells in animal models? And then how can we optimize these cells? So I'll just give you a few kind of summary snippets. So I'm sure everybody in the room knows that, like, our brain has endogenous neural stem cells. So even though it's been felt that that there is no potential for regeneration of plasticity in the adult brain, we now recognize that this can occur. But this is particularly of relevance to the developing brain. So there was a discovery by Sam Weiss in Canada about 15 years ago that there were neural stem cells that have been validated by people like Fred Gage and others, and they're located in the hippocampus and the periventricular zone. And one can isolate these using what's called a neurosphere assay. So here I've just shown in cells that we derive from green fluorescent protein mice. We can isolate these in culture using what's called a neurosphere assay. So we can identify these, and then we can essentially derive any type of neural cell that we want. So we took advantage of this, and this is work that I did in collaboration with Derek Vanderkoy, who's a developmental biologist. And Derek discovered something called the default pathway. So it turns out that in development, embryonic stem cells want to make neurons just by default. You actually don't need to do very much. Well, you need to do a little bit. You need to apply a leukemia inhibitory factor, FGF2, in culture, but you can actually make these neurospheres. And this is really important because everybody else was going through embryonic body formation. And that can be dangerous because you don't want to have mixed cultures with embryonic stem cells and with neural precursor cells, because the ES cells can make teratomas. So when you're injecting the cells into human patients, you want to make sure that you got a really pure population of committed adult neural precursor cells. And then you derive these with a neurosphere assay, so you can actually physically take these cells. You don't have to rely on antibody sorting procedures. So it's a really cool and very simple approach that has the potential for higher throughput. And so we tested this, and this is kind of one of the first proofs of concept we did, and we published this about a decade ago in the Journal of Neuroscience. And this is beta-3 tubulin, just to show the spinal cord. We've injected these above and below the injury site, and these GFP-labeled cells love the white matter niche, and they migrate up and down the spinal cord really well. And they particularly like to make oligodendrocytes. And so here are the stem cells here from YFP mice. They're making myelin, use myelin basic protein, the merged image. And then these are some higher power images. There's an example of a stem cell myelinating one of these axons. So from this, we kind of came up with the following concept. I like movies, so I'll show you another movie. Okay, so here we're showing the spinal cord. Myelinated axons. As you know, each oligodendrocyte typically myelinates 4 to 5 axons. The node of Ranvier, we have the clusters of sodium channels. The myelin is attached to the axon through various adhesion molecules, including Casper. And then next door to this, in the juxtaperonodal region, you have various types of voltage-gated potassium channels. So the molecular organization of the node of Ranvier is extremely important. So after traumatic spinal cord injury, as I've shown you, you have central hemorrhagic necrosis that occurs, and typically you have some sparing of the subpial rim of white matter. If you look closely here, you get secondary cell death of the oligodendrocytes, and as a result, you lose the myelin. There's limited remyelination. And so you have demyelinated sections of the axons, and it's known that in case of demyelination, you have a reorganization of the nodes of Ranvier, and specifically the potassium channels migrate into the nodes, and you have axonal conduction block. So the strategy that we and others are now moving forward, which is going into clinical trials, is using the patient's own spinal cord as an intrinsic scaffold and taking advantage of the fact that the neural precursor cells like the white matter niche, and they migrate along the white matter niche, and they find these denuded axons, remyelinate these axons, and you get a reorganization of the nodes of Ranvier. So now, how do we think that we might try to apply this in a clinical sense? Could we get autologous sources of these? And so here we're taking advantage of the discovery by Yamanaka, who received the Nobel Prize a few years ago, the discovery of induced pluripotent stem cells, where one can introduce transcription factors into somatic cells, and you can make induced pluripotent cells. The problem with the Yamanaka approach, though, is it's relied on viruses, and that has the potential of inducing karyotypic instability. So this is a picture of Andras Nagy, who's one of my collaborators in Toronto, and Andras had a really cool approach that he reported in Nature a few years ago, where he was using a non-viral technique and using what's called a piggyback transposition technique to introduce the Yamanaka transcription factors, and you can remove the piggyback transposon cassette and you have no genetic signature at all. And so this is essentially how we do this in cartoon form. So we introduce the Yamanaka transcription factors, we remove the piggyback transposon cassette, we then use the default pathway, and we get absolutely pure populations of committed adult neural precursor cells. Now we had to figure out how to make a committed adult neural precursor cells from iPS cells. So one of the things is that when we study stem cells, we try to make them fail first. So I can make tumors out of anything, and you really want to do that, right? Because then you figure out how it fails, and then you figure out how to make it fail safe. And so we found that the iPS cells are really dangerous. You know, we were really good at making teratomas out of these cells, but it turns out that you can drive these to a committed adult neural precursor cell type by induction of the Notch signaling pathway. This is a way to inhibit BMP signaling. So by inhibiting BMP signaling, you can drive these along committed neural precursor stage. And here we just showed that we had a downregulation of the primitive marker nest and upregulation of some mature markers, including GFAP, with this. And then we found that the iPS NPCs, when we introduced in the injured spinal cord, they could contribute to remyelination and functional recovery. So here are just some photos here where they're making myelin, and they're myelinating the axons. This is an immuno-EM. So here they've made the myelin, and these little areas here are staining. Whoops. Here, and then we found significant functional recovery. So then the next step was, okay, how do we make a model of cervical injuries? Because that's where the big need is. That's where the trials are. So we came up with this approach. So this was taking advantage of some work that I'd done in my PhD. So we have a unique, this is an aneurysm clip, and it's got a special device to calibrate this for the force. And then we figured out ways to do the injury. This is what the lesion looks like. Very similar to a human lesion. We can image this on an MRI. It's a T2-weighted MRI, and we have very, very clear deficits in forelimb function. So it's a very beautiful model, and we figured out how to do this with very high survival. So we now have close to 100% survival in these animals. So then we went ahead and we applied the neural stem cells in this model. This was an MD PhD student, Jared Wilcox. So he found that he got very nice survival. In this model, we inject them above and below the injury site, and we take advantage of the fact that these stem cells can migrate in the white matter niche. And he found that there was nice survival of the cells. We got a significant reduction in the lesion size, and nice behavioral and electrophysiologic recovery. And the next thing is, okay, how do we study the human cells? And if you just put the human cells into the animal models, you get next to no survival. If you give cyclosporin, which is a commonly given immunosuppressive drug, it's not a lot better. You get like 2% survival. If you use tacrolimus, which is a rather toxic immunosuppressive drug, you get pretty good survival, but the animals are pretty sick with this. So we started using this RNU rat, which is a T-cell deficient athymic nude rat, and we found that we got about the same survival as tacrolimus. And these animals are actually quite healthy. So this is how we do the injury, and this is where we introduce the stem cells above and below the injury. So then the other challenges, though, with the stem cells is how do you promote their neuronal integration and connectivity? How do you drive remyelination? And how do you deal with relatively poor survival? So some of the strategies that I'll just kind of hint at, and this is now some of this work is unpublished, which I'll share with you. So we've been figuring out ways to drive them along a neurogenic line, and we call these neuroectodermal precursor cells. We can make oligogenic neural precursor cells, and we're now looking at ways to introduce growth factors, including ways to introduce glial-derived neural growth factor using a similar non-viral piggyback transposon cassette technique. So this is some actual data. So this is what human iPSC cells look like in culture, then reprogramming them with the piggyback transposon approach. These are the transcription factors that we introduce, and using Andresh's approach. We then use what's called a dual SMAD inhibition approach, where we inhibit BMP as well as TGF-beta signaling, and this drives these along a committed neural precursor cell line. And then now we play games in culture, and I don't know the optimal cell type to look at, so we're looking at various ones. So we can look at neurogenic neural precursor cells, oligogenic neural precursor cells, and then we're also looking at neural precursor cells which are driven to express glial-derived neurotrophic factor. So here we're just showing the pro-OPCs and the NECs, and this is these are stained in green, so these are all stem cells. And here we've just outlined the cavity, and we see that with the pro-OPCs we get slightly better cell survival. Now, this is busy. This is just to show that we get the full array of neural lineage derived from this, but with the pro-OPCs, we get more of an oligogenic lineage. So APC olig2 are oligodendroglial markers, and then with the NEC, we see new N2H1, which are neuronal markers. And both neural ectodermal cells and pro-OPCs contribute to tissue preservation, but it seems that we have the best degree of tissue preservation with the NECs. This is a behavioral assay called the catwalk test, and the catwalk is where the animals walk on a plate of glass. We shine a light, and we can track their paw positioning with a computer. So we can get a computer display of this, and we get over 50 gait parameters. So it's extremely objective, and we see improvements in forelimb swing speed and stride length, as well as improvements in forelimb function in these animals. And then we've been now experimenting with ways to introduce GDNF to try to activate anti-apoptotic pathways and pro-survival genes, including SOX10 and ACT3. And this is just kind of what they look like in culture. So we see improved numbers of GFP-positive cells with GDNF. And this is just some very preliminary work, and this is looking at motor neurons. This is a sham animal, lots of motor neurons in the anterior horn cell. It's an animal with vehicle. We see a drop-off, and then with the stem cell, some preservation with GDNF, a lot more survival. And we think that this may represent trophic factor support, but we need to do a lot more work to confirm this. We also see enhanced tissue sparing and improvement in functional recovery with this approach. So there's a lot of work that's going on. So now what's happening in the clinic, though, is a lot simpler. So currently what's happening in the clinic are allogeneic sources of neural precursor cells without any of these fancy modifications. And this is a reality. You have to start somewhere in the clinical trials. And so some of you may have heard about the well-publicized Geron trial. And then this company kind of crashed and burned because of the huge expenses involved in starting the trial. But there are other trials that have now been launched, including another California-based company called Stem Cell Incorporated. And we're involved with this. So we did the first patients in the world in Zurich, Toronto, and Calgary, and we're now just about to launch a phase 2 randomized trial in cervical spinal cord injury. So just a couple of final messages in the last few minutes. So one of the challenges here also is the glial scar. And one of the strategies that's being looked at is taking advantage of James Fawcett's work with chondroitinase, which is an enzyme which degrades the glial scar. So this is immunohistochemistry showing upregulation of chondroitin sulfide proteoglycans, and these are expressed in the glial scar, and these are felt to be inhibitory to plasticity, and one can significantly degrade these with chondroitinase. We're looking at this with chondroitinase, but we're also starting to look at this with biomaterials. And this is work that we started with Jack Kessler at Northwestern using what are called self-assembling peptides. And these are really cool. This is in cartoon form. This is actually scanning EM, and they come in a liquid form. They assemble at body temperature, and they form a lattice work that looks very much like the extracellular matrix. And so we thought that this might be conducive to promoting repair and regeneration, and might also stimulate neural stem cells to do this. It turns out that these are anti-inflammatory, so we see a reduction in the glial scar, as shown by a reduction in GFAP, and we also see enhanced plasticity. So this is track tracing, where we've introduced a BDA track tracer in the motor cortex, and we're tracing the corticospinal tract, and we see that there's less dieback and there's enhancement of the corticospinal tract. And here we've introduced a tracer distal to the injury, and we're tracing neurons into the brainstem. And so we see that seems to be an improvement in the number of intact axons. So this is something that's still at the late stage, a preclinical level. Now, the last story I'm just going to share with you is what I think is going to be an exciting phase 3 randomized trial, and this is looking at this pathway. And so this is a cartoon of a neuron, and it expresses a receptor called the NGR receptor, and this is really simplified. When the NGR receptor is stimulated by a variety of molecules that are expressed on the inner leaflets of the myelin, including NOGO, oligodendrocyte myelin glycoprotein MAG, it activates a small GTPase called Rho. And when Rho is activated, it can induce some cell death pathways, but the main mechanism is it depolymerizes the actin cytoskeleton. And so when the growth cone, which is initially coming up from the neuron, retracts, and you get a failure of regeneration. So this is work that I moved forward with basic scientist at the University of Montreal, Lisa McCarricker, and I stayed in academia, and Lisa went into business. And Lisa founded a company called Biomaterials, and I made the decision that I wasn't going to invest in Biomaterials. I don't know if that was a mistake or not, but I thought, okay, you know what, I want to do the science, and you get rich. And so the product that's been developed is Sethrin. It's a recombinant Rho GTPase inhibitor, and the way this is applied is at the time of surgery. So you now see the benefits of decompressing the spinal cord. We had access to the human spinal cord, and we can actually introduce this topically to the spinal cord at the time of surgery. It's really cool. And the delivery vehicle is a fibrin sealant, which is actually used by neurosurgeons all the time to seal damaged tissues. And we did this early phase trial, phase 1/2 trial. And so where we've kind of evolved, as you can see, we did all the basic science in the 80s and the 90s, a lot of the phase 1 trials 10 years ago, and now we're moving into phase 3 trials. And what we found here, this is a dose-finding study, cervical and thoracic, and the best results were in the cervical group, and we seem to have found a sweet spot at 3 milligrams, where we found very promising rates of neurological recovery in these subjects. So Lisa's IP has been taken over by a small biotech company called Vertex Pharmaceuticals, and the phase 3 trial, we've actually had the official launch. We haven't enrolled any patients. We just did the official launch 2 weeks ago. So I've kind of taken you on a little bit of a whirlwind tour. I've talked to you a little bit about my own story through some clinical vignettes. We've talked about some clinical issues and some neuroprotective approaches, some ways to stimulate the underlying brain and spinal cord, and then on the last part of the talk about neural stem cells, biomaterials, and biologics. So it's an exciting era, and I think there's lots of opportunity for lateral translation into other forms of central nervous system injury and disease. And then the final note is that would love to see all of you up in Toronto, and the International Neurotrauma Symposium will be up in Toronto in 2018. It should be kind of a cool meeting. And on that note, I'll close, and thank you very much.